Right. About the reactions, we say that it is made up of about 10 steps catalyzed by 10 different enzymes if it is to be aerobic in the presence of oxygen. This is a summary of the whole reactions occurring in glycolysis. First thing is phosphorylation reaction, which is the reaction 1, reaction 3, reaction 7, reaction 10. Then the major thing I want you to know is that whenever you hear phosphorylation, is simply removal of a phosphate or addition of a phosphate by an enzyme referred to as a kinase. In the course of the video, you'll be hearing something like hexokinase, glucokinase, pyruvate kinase, phosphofructokinase 1. Then talk about the rate limiting step or the rate limiting reaction. This is just the step that without that particular one, without the particular step, nothing will occur in glycolysis. If it is inhibited, glycolysis will no longer occur. And also, it is this rate limiting step that differentiates glycolysis from another pathway which is referred to as gluconeogenesis, which is synthesizing glucose from the scratch, starting from its breakdown, which is pyruvate, or starting from any other molecule to give you glucose up. And then you have reaction 1, reaction 3, reaction 10. If you looked at the diagram of what I did, that step, you see those ones on red marker depicts the rate limiting step, which is 1, 3, 10. Then they are catalyzed by these enzymes. Talk about glucokinase, talk about phosphofructokinase 1, talk about pyruvate kinase. These are the 3 enzymes that catalyze the reactions of the rate limiting step in glycolysis. Then for substrate level phosphorylation, what is substrate level phosphorylation? The term is big. Let's try to break it down. Whenever you hear a substrate is a molecule or a compound or an atom, anything that is present within this glycolytic pathway that are referred to as substrate. Then substrate level phosphorylation is just a quicker way of producing ATP instead of going through the oxidative pathway, which is that one occurring within the mitochondria. Then substrate level phosphorylation, people that know grammar, we say that it is the process of transferring phosphate in form of inorganic phosphate from a high energy compound. Example, it can be the one that we know, GTP, which is guanosine triphosphate. You transfer a phosphate from him to a low energy compound, which can be an ADP. If you transfer a phosphate in form of inorganic phosphate to ADP, producing a low energy compound, which will be now, this one will transfer a phosphate to this one in form of inorganic phosphate. It will give us GDP, then this one will come out as what? ATP. This is referred to as substrate level phosphorylation. Process of transferring phosphate, more of inorganic phosphate, from a high energy compound, example GTP, to a low energy compound, example ADP, to produce a low energy compound, respectively, and a high energy compound, ATP. Then the substrate level phosphorylation in glycolysis is simply the reaction 7 and reaction 10. Reaction 7, that is when we are transferring a phosphate from a bisphosphate something to another molecule to form ATP. Then in reaction 10, that time phosphoenol pyruvate is transferring a phosphate to ADP to form ATP to produce another molecule of ATP. That is faster way of producing ATP, which is substrate level phosphorylation. And the fourth reaction is oxido-reduction reaction. The only oxido-reduction reaction is reaction 4. Whenever you hear oxido-reduction, a molecule is oxidized and at the same time it is reduced. The one that is reduced in ATP and oxidized at the same time, it is NAD+. NAD+ is reduced to give us NADH. Remember, I said that NADH is equivalent to how many ATP? 3 ATP, or some people will say it's equivalent to 2.5 ATP. So the only oxido reduction reaction is reaction 6. which is reducing NAD plus to give us NADH. Then second to the last, isomerization reaction, which is the one that converts a molecule to its isomer. Example, glucose 6-phosphate was converted into its isomer, which is fructose 6-phosphate in reaction 2 by an enzyme referred to as an isomerase. Then reaction 5, an isomerase again converted that 3-carbon molecule, which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate into, for instance, that dihydroxyacetone phosphate was converted into glyceraldehyde 3-phosphate by an enzyme referred to as an isomerase. Therefore, that is why I say that isomerization reaction is reaction 2 and 5. Then last but not the least is dehydration reaction. Whenever you hear dehydration, simply means removal of water, and it occurred in reaction 9, which is the one that we removed water from a molecule to produce phosphoenol pyruvate, the one referred to as PEP. Then this reaction is catalyzed by an enolase, dehydration, removal of water. Then this is the summary of the reactions of glycolysis. So in the next one, we are going to be picking them one after the other. Thank you very much.